Welcome to the Aquarium of the Pacific. My name is Sandy Troutwine, and I'm the Vice President of Animal Husbandry here at the Aquarium. First off, I'd like to say thank you to our lecture sponsors, the Gazette newspapers and Courtyard Marriott. And I'd like to everyone to please silence their cell phones during the lecture. Tonight, I am honored to introduce two dear friends and colleagues, Nate Jaros and Briana Fodor. Tonight, they will discuss the aquarium's important work in coral reef restoration in Palau and the Bahamas. Briana and Nate are both husbandry staff members here at the aquarium. Briana is a senior aquarist, and Nate is the aquarium's curator of fish and invertebrates. Last year, Nate traveled to Palau to support SECORE International and the Newcastle University's CORALASSIST lab. This work included aiding in the research to study the effects of warming seas on coral populations and techniques for scalable restoration methods. Nate previously worked as an aquarist at the Henry Dorley Zoo in Omaha, Nebraska, and then we stole him, and he's been working at the Aquarium of the Pacific since 2005. He started as an aquarist here at the aquarium, and he focused originally on seahorse reproduction, sea jellies, and other species, and has skilled in many, many other things, including building our own hyperbaric chamber here at the aquarium. Briana's career at the aquarium began as an aquarist intern in 2009, shortly after I had the pleasure of meeting Briana through a marine biology quarter field excursion through UCLA. I remember meeting her in Moorea, in French Polynesia, actually, and her passion for marine biology was evident then, and we were so delighted when she became a full-time aquarist in 2010. She's spent 6 years working with the aquarium's live tropical coral exhibits. She's used her experience as a certified scientific diver and knowledge of corals on her trip to the Bahamas in 2019 to start as part of a SECORE workshop on coral restoration. Tonight, Nate and Briana will be talking to us about coral reef restoration in Palau and the Bahamas. Welcome, Nate and Briana.
Hello, everybody. Welcome. Thanks for coming. We got a good turnout tonight. Looks like there's a big group. Thanks for joining us. We'd like to start by thanking you for coming and for your support of the aquarium. Tonight we're going to review coral reef restoration efforts, projects that the AOP is directly related to, and a few that aren't. We'll highlight our efforts with SECORE International, as Sandy mentioned. But we'll also review kind of a tight-knit network of different agencies, government agencies, universities, NGOs, and aquariums and zoos that work together to try to help work a common goal towards saving coral reefs. So I'm obligated to start with a certain amount of information just to kind of give an overview of the threats to coral reefs. This quote kind of summarizes that climate change is the greatest global threat to coral reef ecosystem. Scientific evidence now clearly indicates that the Earth's atmosphere and oceans are warming and that these changes are primarily due to greenhouse gases derived from human activities. So I'm going to have to go through a series of slides, obligatory, if we're going to talk about coral reef restoration. A lot of you are probably familiar with some of these slides. There are a lot of threats to coral reefs, including pollution, toxins, coastal development. The list goes on, but the coral reefs are resilient. They can bounce back from different threats, and they can come back to and do quite well. But when the pressures or the stressors are sustained for extended periods, that's when we're in real trouble, and that's why climate change is a serious one. So this is just a graph showing greenhouse gas, carbon dioxide accumulation over time, and you can see that in our current state, we as the whole world is producing about 30 billion metric tons per year of carbon dioxide. These fossil fuels are greenhouse gases that warm the atmosphere over time. Again, this is just a graph that shows the temperature rise over time, and you can see that throughout the the 20th century and into the 21st century, the temperature has just gone up and up, with it peaking in 2016. We've gone up 2 full degrees Fahrenheit since 1880, and this has serious effects on the ocean and sea level, our global ice. So, where's the heat going? The oceans absorb an incredible amount of heat energy. The vertical of this graph is showing numbers in zettajoules. 1 zettajoule is enough to power the equivalent of 25 billion American households for a year. The ocean, as you can see, absorbs almost all of that heat energy from the earth. We're very lucky that we have such a massive ocean system. Otherwise, we would have been in trouble a long time ago. But this isn't the capacity isn't unlimited. This could create problems if we continue to warm the atmosphere, because although the ocean is currently absorbing it, it's not always going to be the case. And this has serious implications to weather patterns. We see more violent storms, more flooding, more rain because of the warmer ocean. There it is. So the greenhouse gases, as I mentioned, are mostly carbon dioxide. The carbon dioxide goes into the atmosphere from anthropogenic sources, and the ocean absorbs a lot of them. It absorbs about 25% of the carbon dioxide that we pump into the atmosphere. Yet another wonderful thing that the ocean can do, it can absorb a lot of that in addition to the heat. A large percentage of it will cycle back into the atmosphere, and about a huge amount will go down into the deep sea. The deep ocean concentration of carbon dioxide is about 50 times greater than that in the atmosphere. And the trouble with this is, as dissolved carbon dioxide increases in the ocean, it creates a process called ocean acidification, which, as the dissolved carbon dioxide goes up, the pH goes down, which can have a lot of serious implications to animals, largely corals. Corals use dissolved calcium and calcium carbonate to build their skeletons. So as that pH decreases, that's going to be very bad for them. The skeletons are weaker, or they're slower to form, creating stress on the corals. I'm going to hand it off to Briana. She's going to talk a little bit more about coral biology at this point. Go ahead.
All right. So before we get too much into it, I figured I'd just explain a little bit what is actually a coral. So corals are, in fact, animals. They're related to animals such as sea jellies and sea anemones. They're in the phylum Cnidaria. Let's see, you're here. They are found all over the world, from the shallow tropical warm reefs all the way down to really deep, dark, cold areas of the oceans. They are very slow growing, averaging only about 2 to 10 centimeters per year. And they really do come in all shapes and sizes. Everything from a single colony or a single polyp to a huge colony that could grow as big as a car. Some of them have soft bodies, some of them are hard, as Nate was saying, and make their own skeletons. But coral reefs are, I think I'm going too fast. Where am I? Okay. So corals are the foundations for coral reefs. They encourage high animal diversity by providing homes, hiding spots, and even food for other animals on the reef. This allows corals to be one of the most diverse ecosystems on the planet. Coral reefs are also important for humans. Coral reefs provide food, protection from high surf, and ecotourism jobs for humans living in communities near them. They also contribute to healthy fisheries worldwide and are important places for medical research. Sorry, getting used to all the buttons. Give me a second. Yeah, and here, I mean, obviously very beautiful places, very diverse ecosystem, so I figure I'll show you guys some pretty pictures of very healthy coral reefs. All right, so a little bit about coral reproduction, which is a very important part with coral restoration, which we'll get to in a little bit. But first, some species of corals are what's called gonochoric. That means that colonies are different sexes. So there's male corals and there's female corals. Some types of corals are hermaphroditic. This means one single colony can be both male and female. Corals will have a mass spawning event where corals of the same species will all release their gametes on the same time, the same night. This is based on changes in water temperature, lunar cycles, and as well as the time after sunset. And then here's just a little life cycle map for you guys. But corals are also able to produce asexually. One of these is called budding. It's the picture on the left. So a coral colony is able to drop off a little miniature clone of itself. This little clone will go find itself a spot on the reef and attach and become a whole new coral. Corals are also able to reproduce through fragmentation. This is either when a piece of a coral breaks off either from a storm, a clumsy diver, but then this coral can go find it and again attach to the reef and again become a new coral. All right, so corals have polyps, which you can see diagrammed over in the right diagram there. These are used to help capture food as well aid in their reproduction. Many tropical corals also have a symbiotic algae that live in their tissue. This is called zooxanthellae. Photosynthetic corals that have zooxanthellae require these to survive. What the algae does is it uses waste from the corals in order to aid in photosynthesis, and then in turn, the algae provides the coral with sugars and amino acids. So now if these zooxanthellae aren't happy in the coral for some reason, whether it's from maybe warming temperatures, it could be pollutants in the water, the zooxanthellae will leave the coral and its coral hosts and what we term coral bleaching. So these photosynthetic corals, like I was saying earlier, if they don't have zooxanthellae in their tissues and they turn this white bleach color, the animal itself might still be alive, but it won't last long. They aren't able to catch enough food in the water column for themselves to live, so they do need those zooxanthellae to survive. So here's a picture of an area. So this was a coral reef. Actually, I think it was Alpha Morea, like Sandy had mentioned earlier. And unfortunately, all the white colonies in here that look really white and clean are bleached corals. So they're ones where their zooxanthellae have left their tissue. And you can see them in contrast to the corals that are still alive. They're the little bit more browner colored corals in there. So in a mass bleaching where all corals bleach and eventually die on the reef, entire reefs can potentially be destroyed, changing the landscape of the underwater environment. This in turn can lead to a less healthy reef and decreased animal diversity. So like Nate had kind of mentioned earlier, coral reefs are historically can be quite resilient, based on all the indicators that are listed above. So they may be initially damaged by a major event, such as coral diseases, coral bleaching, a large storm, or even a major predatory event. And the coral reefs themselves are able to come back from it. However, currently, because of the warmer temperatures and the ocean acidification that Nate had described, it's making it a lot more difficult for these coral reefs to be able to handle any kind of a disturbance like that. So we're at the point now where many of our coral scientists have decided that it's time for, humans to step in and really to help restore some of these coral reefs that are really being strongly affected.
Thanks, Brianna. So, reef restoration. Thank you. We'll switch a few more times. We can save the applause till the end. Reef restoration. I just want to kind of talk about what that is. So, reef restoration is research and conservation work to study, protect, and restore coral reefs. But why do it? As Brianna said, bleaching events, if reefs are destroyed by storms or pollution, et cetera, the list goes on. Who does it? Government agencies, non-government agencies, zoos, aquariums, AZA, which is the governing body of zoos and aquariums, which we'll get into in a minute. And there's a lot of different avenues and agencies that get involved with this and a lot of different partnerships that work together. Where is it happening? pretty much all of the tropical seas where there's corals. There's work being done in the Florida Atlantic, in the Great Barrier Reef, the Caribbean, Hawaii, and the Indo-Pacific. And when? Hopefully before the unnecessary loss of species. I think there's a lot being done on the fronts of where the species of individual species are really being diminished. So this is an example of a decision matrix from a coral reef restoration publication. I don't intend for you to review and absorb all that information. The point I put it up here is that not every reef in the ocean meets the qualifications to put effort into restoration. If you just read the top box, it says, have the agents of degradation been permanently removed? And if they haven't, there's not really a reason to put effort, money, and labor into restoring that reef. First, you have to cut off the cause before you can start to treat the symptoms. Other factors are including, of course, I mean, you would only restore a reef in an area where there's historically been a reef, where it meets all the natural parameters for those corals to live there. You wouldn't want to just go out to a sand flat and start a new coral reef. It just doesn't work that way. And economics work into it as well. So I'm going to talk a little bit about SECORE, SECORE International. This is an agency that combines research and active reef restoration, education, and outreach. It's a network of scientists and public aquarium professionals and various stakeholders, and the aim is a scalable restoration effort, which I'll get to in a minute. It was started by Dr. Dirk Peterson. He was the founder and executive director. He started this nearly 20 years ago when he was working at the Rotterdam Zoo. There's a reason that our aquarium and a lot of other public aquariums are tied into this network, because it kind of has its roots in aquariums. There's a lot that aquariums have offered the scientific community, particularly with keeping very challenging animals in captivity like coral. You can learn a lot from keeping animals in aquariums, and we've applied this knowledge and kind of taken it out into the field. So our aquarium and many others help provide funding. We provide boots on the ground out in the field to do some of this restoration work. So Brianna talked about coral spawning. Before I went out on my first spawning dive, this is kind of what I was expecting, the entire reef, this cathartic experience where it's reverse snow from the bottom up. And sometimes you see it, but not every coral on the reef goes to spawn at the same time. Sometimes it's one here or there, one set of species here or there. Sometimes it's a lot of waiting and hours of diving and circling around and just waiting and hoping that you'll see a little bit of gametes being released. But there are these massive, obviously from the photograph, there are these massive spawning events that are quite spectacular. In some cases, the coral will spawn and release gametes from every single polyp at the same time, so it almost looks like the entire shape of the coral is just lifted up off the bottom, and then it starts to disperse with the waves in action. It is a wonderful experience. I don't mean to belittle it, but it's not always quite like that. So SECORE, what they do, and what a lot of other scientists do, is we understand the patterns of these. As Brianna mentioned, they're very cyclical. They do need to spawn at the same time to be effective, so they spawn in a certain number of days after a full moon. We can study that, we can learn, we can keep records, and then you generally go out a few days early just in case there's any variance. But SECORE will make these what we call spawning tents. So if a coral is about to spawn, you can go cover that coral in this fine mesh that won't allow the gametes to pass through and has the floating upside down cup. So when it spawns, the gametes will be collected into that vessel. They're taken back to the lab and fertilized in a controlled setting just to increase the percentage of fertilization, and also settled in a, didn't put the slides up. Fertilization, and they're settled onto what are called substrates or tiles, where the young corals go from a swimming larvae to a settled coral, which will immediately start to asexually divide and have multiple polyps. So they settle onto these substrates, which then can be moved out onto the reef, and they can be moved into an area that qualifies for reef restoration or needs that additional help, or we can spawn individuals that maybe genetically are unique. Particularly if there's a bottleneck in a particular region, we can study the genetics and we can spawn and recruit corals to an area that might be genetically unique, so we can strengthen the population. And then disperse those substrates out in those areas. So looking at this is kind of where scalability and reach come into play. So it's relatively easy to go to a location where there are reefs that has a really robust marine lab, and you can utilize a lot of expensive equipment to do all this work in a lab setting. What SECORE wants to do is, what if a reef needs to be restored and they don't have those amenities? So they've created what's called an in situ settlement pool, which is a fancy name for just kind of a floating boat. And they can put all these larvae, fertilized eggs and larvae, into this vessel. It has screens on it, so ocean water can come, clean ocean water can come in and out, but the larvae can't pass out, and predators can't come in. And in these pools, which can be tied off anywhere where there's clean ocean water, those corals can develop onto these tiles and start to grow, and they can just stay here in protection until they're examined and determined that the corals are at a state where they're ready to be outplanted. So in one picture you can see into the pool, there's lots of different trays with all these different settlement tiles, and the other one you can see it just kind of from above. They went through a lot of different iterations of this design, and the one thing that they found is that it just needs to be weatherproof. It's designed kind of like a boat with pointy ends, so if waves or currents are coming, it can kind of deflect a lot of that energy so they don't get torn up by the environmental conditions. They actually have a shade structure that you can plant on top, because even though these are corals that need light, they're very close to the surface and they're very small corals, so they kind of need to be in a protected environment. If they were to settle on the reef, they may settle in slightly deeper water or underside of a rock or something like that. So kind of mimic the conditions that are ideal for settling and survival. And then once they're ready, they can be taken out and outplanted. You can see this diver has one of these substrates or settlement tiles in her hand and a basket full of more, and she can quickly go about the reef and plant those tiles out there. Early restoration and research efforts, this isn't novel. You can settle coral larvae onto small tiles, but in some cases you'd go with an underwater drill. You'd drill a hole, you'd use epoxy, you'd attach that small tile one at a time just to study the effectiveness of the restoration efforts, but that takes way too much time if you're talking about building up a reef. The Great Barrier Reef is about 1,500 miles long. To be drilling individual corals in time of need is not going to be very effective. So this organization is looking at scalability. How can you outplant a lot of corals? If you had millions of these tiles, could they be scattered from a boat? And even if a small percentage landed in a place where they could grow into a new colony, would it be more effective than trying to individually plant a coral one at a time? One of the partners of SECORE International that, as Sandy mentioned, that I worked with in Palau was CORALASSIST. This is the university research side, studying the effectiveness of restoration out of Newcastle University. And they use assisted gene flow and selective breeding and adaptation to study corals, led by Dr. James Guest. I kind of mention this because this is an important front of reef restoration, particularly in the face of warming ocean seas, ocean temperature. We can look to corals that are naturally resilient. In some cases, you'll go to a reef and one coral of one species and another one of the same species in the exact same conditions. One bleaches and dies and the other one doesn't, because there's something about that coral that's more adaptive or more resilient. So there's a lot of people that are studying those corals, and this particular university isn't just studying them, they're crossing them and looking at different things. So they're doing a 5-year project in Palau. I think they're in their 3rd year currently. And they basically take small samples, fragments as we call them, or frags, from a healthy reef, identify them, and put them into these controlled baths and subject them to different temperatures in a controlled setting, and which ones can tolerate it, which ones can't. And once those have been identified, you can see in this picture some are white and some are not. So they've actually tracked this individual colony and where it is on the reef, and this one as well. So this would be called low tolerance and high tolerance. And they go, when it's spawning time, they actually take a small colony and bring it into the aquarium and spawn it in a controlled setting. So as opposed to the SECORE method where we take a big tent out at night, and sometimes there's a lot of surge, and when those corals spawn, so these worms come out and they're getting all in your ears and face, it's disgusting. They put them in an aquarium, have a cup of coffee, and just wait for the spawn to trickle out, collect it all, and get fertilization in these specialty tanks and grow up the young individuals. So it's a 5-year study. So they've got these crosses. They've got high tolerance to low tolerance, high to medium, medium to low, and every single combination of those, so they can study and get an effective data to make some really informed decisions about reef restoration. To grow those up, they're going to grow those in nurseries out in the ocean, and they're going to grow them to the point where they can be spawned, and then they can do additional crosses and just be really thorough with what the limitations are for selective, as I say, just assisted gene flow in coral restoration. Just a brief mention of Gates' Coral Lab. Ruth Gates, she gave a presentation here a few years ago, and unfortunately she passed away, but despite the loss of this wonderful and inspirational person, her lab continues on in her research, and she's doing a lot of, that lab is doing a lot of the same type of work. In Hawaii, there's been, in 2019, there was a bleaching event making 3 in the last 5 years, and despite the unfortunate loss of Ruth Gates, her team continues her research and vision, utilizing technology, looking at different symbionts within algae tissues and the zooxanthellae and the microbiome. They're just really taking a lot of different angles to what's resilient and what's not, and using some of the same selective breeding techniques to try to help really local populations in Hawaii. This is their lab, which would be a wonderful place to do it. It looks like we have a lot of students out in the audience. If you want to go do a specialty coral project, could you imagine working at this place? And then just to mention another, you'll kind of see a theme. The Australian Institute of Marine Sciences is a government agency. They're doing a lot of great restoration and research work on the Great Barrier Reef. At this point, I'm not going to go on and on about some of the same types of things that they're doing, but one unique thing they have is this picture is taken from a boat. They basically built an enormous marine lab that can go out to where the reef is and eliminate a lot of the time involved in research and cut out the middleman, and they can do experimentation and restoration work right there on the vessel. So Brianna's coming back to the lectern.
All right, so I'm just going to talk about a few other coral restoration efforts, mostly on the east coast of the U.S. One important one is involving the Florida Reef Tract. This is the largest and actually the only barrier reef in the continental U.S. It is actually the 3rd largest barrier reef in the entire world. But since about 2014, the corals along this barrier reef have been experiencing a substantial disease-related die-off of multiple species of corals all over these reefs. So one kind of interesting coral restoration effort that's currently occurring is the AZA Florida Reef Tract Rescue Project. This was established in November 2018. This project is involving actually aquariums and zoos all over the country. So what's happening is they are collecting 22 colonies of 22 different species of coral that are on the Florida reefs, and the corals that are still healthy at least. And these are being transported into aquariums. And then these aquariums are now responsible for caring for these corals. And what they're trying to do is create a coral ark. So hopefully in the future, once the Florida Reef Tract is safe again for corals, we can outplant these corals, put them back on reefs, and then kind of start over and create a new coral reef. Another restoration project is the Coral Restoration Foundation. These guys are working on 11 different species, again, of corals on that reef tract. And what they do are they use these coral tree nurseries that are pictured here. So they'll take fragments of healthy corals, hang them on what they call coral trees, and raise them up into larger colonies, and then they'll put those colonies back on healthy reefs. Since 2007, more than 100,000 corals have been planted back onto the reefs by the Coral Restoration Foundation. So now I'm going to talk a little bit about the Bahamas. As of 2016, living corals only covered 10 to 12% of the Bahamian reefs. And this is a result of widespread bleaching events, mostly caused by warming water temperatures, but also from disease outbreaks. So Vita is one of the companies doing coral restoration on the coral reefs. What these guys are doing is they use land-based coral farms in order to raise corals to a larger size that they can then outplant on the reefs. And they use something that's kind of interesting. It's called microfragging, or microfragmenting. So what they'll do is they'll take a larger coral, break it up into smaller pieces, and you can see it kind of on the right side. They'll grow these corals next to each other. And from there, the coral will want to bridge the gap between each of its fragments. So in the end, it's going to create a larger coral much quicker than if you just had that first original coral and letting it grow to a bigger size. And then again, these can be outplanted back onto reefs. They're also working on creating, again, more resilient corals to the changing ocean conditions by altering the water parameters in the tanks that they're raising corals in. So the Bahamas Coral Innovation Hub is a group of various organizations. This includes the Nature Conservancy, the Perry Institute for Marine Science, SECORE International, and Cape Eleuthera Institute. They are using the methods of SECORE, so using sexual reproduction in order to, again, help with coral restoration, but also to perform research on creating more hardy corals for, like I said, the changing water. All right, well, I'm going to get started kind of talking about my trip into SECORE. So back in September of 2019, I was fortunate enough to be asked to go to a SECORE coral spawning workshop in the Bahamas. It was located on the island of Eleuthera. And for those of you who know where Eleuthera is, which I didn't know until I went to this workshop, it's that tiny little island in the Bahamas. And it's kind of interesting. It's a very small island. It's 110 miles long. And at many parts of the island, it's less than a mile wide, so very skinny as well. This workshop was hosted by Cape Eleuthera Institute, which is on the more southern side of Eleuthera. And we had 3 SECORE representatives, 3 scientists from various coral restoration efforts, mostly based out of Florida, and also a staff from Cape Eleuthera who hosted this workshop. And they trained 6 aquarists, including me, on the process of using coral spawning in order to reproduce corals for coral restoration. All right, so this is a video of the site that I got to work on when we were there. It's called John Miller's Blue Hole. The reason this location was picked is that there is actually a big blue hole in the center of the reef, and it's a deep hole that has cold water upwelling through it that keeps that reef itself a little bit cooler. Because the temperature is a little bit cooler compared to other reefs, and when I say cool, it's still low 80s. It's very warm compared to things here, but it is a better temperature for coral, so the reefs themselves are really healthy. So this was just a video, like I said, from during our first orientation dive. So on the reef itself, we split the reef up into 4 different zones, and each buddy pair was responsible for surveying a certain zone during the night dives, looking for coral spawning. So we had gone out that first day. You probably saw the water bottle floating in the water. That was actually a marker. We used that at night so that, I'm sure you can imagine, it's pretty dark out there at night, so we could use the water bottles to kind of as a guide to see where we were within the reef. And then in each of these zones, we had certain coral colonies that were numbered and tagged. This was done before we arrived in Eleuthera. And these were corals, all the same species. It's Orbicella faviolata. And they were ones that they wanted us to keep a kind of a closer eye on. Their plan is to monitor these corals throughout the years as they're doing these spawning efforts, just to monitor coral health, success with spawning. We made sure to kind of see those as we were doing just loops throughout the reef looking for spawn. So we were very successful. We collected gametes from multiple colonies off of these night dives, and were able to produce a lot of larvae. The location that we were at was about an hour boat ride from where we were staying and the lab that we were working in. So we would fertilize the gametes on the boat, bring back with us, and then from there we could clean them back in the lab better. And then a few days later, we were able to settle them onto those substrates that Nate had kind of shown pictures of earlier. So I'm sure you can see, you may recognize we have the pools again where we would have substrates. And and then a big thing that SECORE is working on in Eleuthera is they're mostly focusing on just ways to improve their success rate. So they're looking at picking the right substrate shape, the right substrate composition, and also, again, trying to produce more heat-tolerant coral larvae. So all the larvae that we collected on this trip were used for those experiments that they helped set up. And then those will continue to be monitored by SECORE staff as well as some of the staff at Cape Eleuthera Institute. Great.
Oh no. That picture was supposed to pop up. So that's a world map underneath that picture that shows where Palau is. So let me just describe Palau, because we're going to talk about the spring trip that I went on last year to support SECORE's work. Palau is just a few hundred miles east of the Philippines, just north of the equator, just kind of in the middle of the South Pacific Ocean. Beautiful. It's known for beautiful diving. It's a recreational spot for a lot of world travelers. This is a photo I took on a reef. This is not uncommon to see just schools of beautiful, colorful fish, thriving coral reefs, amazing drop-offs, just fascinating place to be. It's definitely paradise as far as I'm concerned. Here's just kind of the bathymetry around Palau. You can see the red locations are islands. This is the largest island, and it's a volcanic island. And then there's just about 350 to 700, depending what you qualify as an island, just this dense structure of what they call Palau's rock islands, covered in vegetation. These are remnants of ancient coral reefs, limestone, that's changed with plate tectonics. There's a huge barrier reef, about 100 miles long. For World War II buffs, there was a lot of wars, battles fought here. Peleolu is a very famous one, and there's still a lot of World War II ruins, both on land and underwater, that a lot of people go to see in this area. Excuse me. So why use Palau as a destination for coral study? There is just some unique things about Palau. This is deep ocean water. It's not just a drop off of the map. There's an abyssal plain that rises up. This is just a dense network of shallow, warm water lagoons. These lagoons have been warm for a very long time based on just the natural conditions of Palau. So there's a lot of corals that are already kind of were exposed to warmer water temperatures, which makes it an excellent place to study heat tolerance. There's very healthy reefs. If this is an area where there's a lot of research happening, as I said, the reefs are in very good shape. It's not necessarily a candidate for restoration, but if you're studying corals and their behaviors and spawning and doing experimentation and you need to take coral off the reef, you don't want to do that in an area that's in severe need of restoration. You do it in an area that's already very healthy, which makes for a really nice place to work as well. The government is fairly supportive. There's a marine lab on site that has a lot of resources, and an International Coral Reef Center. As mentioned, I went there. I supported the work of SECORE International as well as CORALASSIST out of the UK. Had a wonderful experience, and we'll be sending 2 additional staff who are in the audience tonight in May to continue this work with these folks. So this is just kind of a snapshot of where we did some of our work. There's an aquarium in Palau that shares a campus with the Palau International Coral Reef Center, which they call PICRIC. So we work at PICRIC, and it's actually on a pier that was made by the Japanese Army during World War II. It's a concrete pier. And as I mentioned, there's this dense network of rock islands around Palau. There's actually a channel that was dynamited out into a very secluded bay where this is just wide enough for a boat, and there's a lot of Japanese signs that are remnants of World War II. So we'd take our boat in and out of that secret passageway to the secret marine lab every day, and it was a pretty cool experience. Here's a snapshot of the in situ settlement pool in Palau, just tied right off the pier that we were working on, so you can kind of see the natural beauty of Palau. Reefs everywhere. And here's a snapshot of just kind of what we were doing. We did fertilization in the lab, settled the tiles in the substrate, in the settlement pool, and then when they were ready, we took them out to the reef to outplant them. This is kind of a pilot study. As mentioned, this isn't an area that needs significant restoration, so it's kind of a proof of concept area. So we take these settlement tiles, take them out to the reef. We don't just randomly scatter them. We need to circle back and see how they're doing in year 1 or year 2, year 3, et cetera. Are the corals growing? Are they thriving? Is this a practice that could be employed elsewhere to success? So we lay out a lot of transects or measuring tapes and take diligent notes on where individual tiles are placed so they can be monitored over time. And you can kind of see a close-up of one of these settlement tiles. I didn't really describe it very well. There's a lot of different shapes and sizes that this organization has used over time. And what they really look for are a lot of surface area, lots of either depressions or areas that come off of the base, just to give lots of little nooks and crannies, because corals will find just protected areas in some cases to settle. These tiles are conditioned or left out in the sea to recruit a certain amount of small algae, and coralline algae especially, that can provide settlement cues for the swimming coral larvae to settle on. And their shape is designed, some of them they're called tetrapods, others have 4 or 5 branching arms, and they're just designed to be wedged quickly, as I mentioned, for the scalability into these crevices on the reef. Rather than having to be drilled in and secured. And not everyone is going to stay, not everyone's going to be fruitful, but ideally each one of these settlement tiles will have hundreds, if not thousands, of settled corals of the same species. And the really cool thing about corals is that they can grow together to form larger organisms. So if there's a lot of very small individual polyps that have settled on there and they're in close enough proximity, as they grow, they can just merge to form one large colony. And that was it for Palau.
All right, so as you can see, although many coral reefs are currently at great risk and in danger of disappearing, there are many organizations working to help save them. As you can see, just a few pictures here of various efforts. And even though most of us are not able to directly work with coral restoration groups, there are still many actions we can take to help protect coral reefs at home. By supporting institutions like the Aquarium of the Pacific that assist in coral restoration and other conservation efforts, you in turn are helping to protect our oceans and the valuable ecosystems in them. With that note, I'll leave you guys with this to kind of peruse to see various ways that you can help to be a little bit more green. And I think with that, just thank you all for coming tonight. And I think we have questions. Yeah.
Great job. Thank you. All right, we have some time for questions. I can pass around the microphone. We asked that, since we are filming this for other folks, if you could just speak into the microphone. Questions? Who has a question? Right behind you there.
I was wondering what the tiles themselves are made out of.
Oh, sorry.
There's a lot of different materials that they've used, and they're still refining that. Some of it comes down to functionality, other of it comes down to economics and scalability. But concrete has been a very popular one. The ones that you saw in the photo are ceramic, and they get people that have these types of shops that can create these things, and a lot of them are donating their materials and providing these at no cost for the program.
Sorry if I'm making you repeat yourself, but what was I gonna say? I'm so sorry. I forgot what I was gonna say. I need to think about it for a second. I'm so sorry.
That's all right. Anybody else?
Yeah. Never mind. I'm sorry. Can I raise my hand if I think about it later?
Okay.
I'm sorry.
That's right. We have one here, Linda.
Yes, what are some of the major consequences if we lose the coral reefs?
I had kind of mentioned earlier, but they're just very important, not only to animals, but also to humans. People who live near coral reefs, they provide the major source of food for them. Also, jobs are very important, and they also provide protection from any kind of big surf or swell that come in and, to those smaller islands. And then just globally, they really promote and help with fisheries by giving juvenile fish, especially even ones that we eat, a place to grow up and hide before they go out into the open ocean. And they're also very important for just medical research as well. So.
Yeah, they're also one of the most diverse ecosystems in the world. So a lot of untapped potential. We know of a lot of the medical applications, but there could be others that we don't know about yet, and it would be a shame if some of those things went extinct before we were able to learn.
What would you say is the success rate of the corals that you guys are planting, and the ones that have been successful, how much growth have you guys been seeing?
That's a good question. The fertilization and settlement rate is very high. We can get 80s to 90% fertilization in a controlled setting. There's just that much information out there. Settling is, everything kind of steps down. Settling and then getting them to grow on the substrates is a little bit less. There are some studies. Although this program's 20 years old, it's gone through a lot of different morphs and iterations. So there are a lot of corals that have grown to full spawning size, particularly in the Caribbean, where a lot of their primary research is done, particularly around Curacao. And there's been some wonderful proof of concept information there, but as far as great measurable data, we're not really there yet.
Thank you for your presentation. Regarding the topic you have up there, don't send chemicals into our waterways, what are some of the things that are going into the waters around this area besides fertilizer, maybe prescription drugs? What are some of the things that's destroying the coral reefs? And does it vary depending on the location of the coral reefs? Do they have an immunity that they build against it? Thank you.
Started, at least you can add to it. So, yeah, some of these places where coral reefs are around islands that aren't very developed. So, quite often because that there aren't proper, sewage, things like that. So, like you were saying, fertilizer is big. Just any of that runoff, which in general is going to ruin the water quality where the corals are. And I didn't really mention this, but corals need, for the most part, very pristine, very clear water. So, if those enter the waterways and kind of muck up, I guess, the water where corals live, it could really harm them. So,
no, I don't think I'd add much to that. Sedimentation is a big one. I mean, the sedimentation in big urban areas affects both the water clarity and the light penetration, but it can also settle onto the coral's tissues. Sunscreens up here, did you mention that? Sunscreen, is it going to ruin the planet's corals? Probably not, but I think we can make smart choices and use reef safe, eliminate certain chemicals that we know to be harmful to corals, and just make wise choices. Try to leave little impact on our environment especially when you go visit these areas. But even at home, that's a big question we were talking about earlier is coral reefs. We live here in California. We have corals. A lot of people are surprised to hear that, but we don't have these big, vast tropical coral reef systems. So why does it matter what you do here to affect coral reefs? But it is a global ocean. The choices that you make, the elected officials that you vote for, the amount of carbon that we're putting in the atmosphere, you can really, everybody can do their part.
Maybe one more question? Oh, we've got two. Maybe two more. Hold on.
So seeing the trend and the raising in the temperature that we're seeing right now in the world and the discussion on how this impacts the coral, along with that 5-year study where they're trying to find coral that's more resistant to heat, do you think it could be a situation where the coral that is more resistant to heat survives and ends up decreasing the genetic diversity of coral, essentially crashing coral ecosystems?
That's an excellent question, and that's where a lot of those crosses come in. That's why it's important not to just we toss around the term supercorals. Some researchers coined that phrase. What we really talk about is just tolerance. Corals that are naturally suited to that environment, and then and the heredity of that. If you only selected all of the top corals, you would certainly be limiting genetics. But if you can find that crossing a tolerant coral with an intolerant coral can produce a moderately tolerant coral or a high tolerant coral, that's going to help maintain some of that genetic importance.
Okay, last question.
And piggybacking off her question, is there any way we could geoengineer the corals, like the deep sea gorgonians off our coast, to withstand those temperature changes, or?
That's over my pay grade. But yeah, I'm sure that there are people that are doing work with it, especially with the assisted gene flow. I'm not sure about modifying the genetics, though. Brianna? Sandy?
Well, thank you, Nate and Brianna. Let's give them another round. You certainly inspired us tonight. And I'd just like to reiterate what Nate and Brianna said earlier about there are lots of great opportunities in coral reef research now and conservation, and it's a really big area, a new area. And so I encourage all of you students out there to really kind of look into it, because there are a lot of great opportunities. So thank you for being here tonight. And I did want to say that our next lecture will be February 26th, when Andrew Reeves will be here to discuss the modern environmental movement and the introduction of the invasive Asian carp. If you can't attend in person, please tune in for the live web broadcast. Thank you, and have a good evening.